This video will introduce the topic of complex and quantitative traits. Complex traits are those that are determined by several genes and in many cases also the environment. Many of these traits can be described numerically. The field of quantitative genetics studies the mode of inheritance of complex and quantitative traits. The multiple influences on quantitative traits means that they tend to exhibit a continuum of phenotypes. These include traits such as length, height, and weight, which are termed continuous traits. Some quantitative traits are not continuous and are considered meristic or discrete. Many diseases are thought to be threshold traits, which are inherited due to the contributions of several different genes. The presence and severity of the disease often is based on having at least a minimum number of specific alleles for the genes involved in the particular disease. Typically, we will divide quantitative traits simply into two categories, continuous and discrete. In addition to quantitative traits, some traits are qualitative. These are traits that involve a presence-absence of a feature rather than any sort of measurement. Quantitative traits are any traits that have mathematically measurable units. These can be continuous, meaning that they vary continuously, or discrete traits, which are expressed as whole numbers or fall into specific phenotypic categories. Continuous and quantitative traits are often visualized using frequency distributions. These show the number or proportion of individuals with traits of in particular ranges. Now, a great example of this is human height, which shows a normal distribution with enough samples, meaning most people fall near the middle of the full range of heights that are observed in humans. As stated previously, a reason for the observed continuum in many quantitative traits is due to them being governed by multiple genes. This is termed polygenic inheritance, and specifically, this term refers to traits controlled by two or more different genes. In many cases, like human height, the number of genes that control this trait is very large, along with many often unknown environmental factors. Polygenic inheritance was first demonstrated in wheat in 1909 by Herman Nielsen L. He observed a peculiar pattern when combining true breeding white and red-hulled wheat that was inconsistent with inheritance by a single gene. When he combined his true breeding color variants, the F1 generation yielded an intermediate phenotype. When the F1s were combined, the F2 generation showed this continuum of color from dark red to white in a ratio of 1 to 4 to 6 to 4 to 1. Based on this pattern, he concluded that two different genes were active in controlling hull color and that each locus contributed to the phenotype additively. On top of multiple genes influencing phenotypes, the environment can also play a significant role. In the example here of environmental influence, we see that high environmental effects, shown on the right, can elicit a continuum of phenotypes even under single gene control. When traits are controlled by multiple genes, so more than one gene plus the environment, as shown here with a three gene influence plus environment, it can be difficult to near impossible to categorize them into discrete genotypic and phenotypic categories. And for these traits, we often need to use mathematics to unravel the relative contributions of genetics versus the environment.